All right, so here we're going to get into the function of a protein. And there are a very large number of different protein functions, and we can go into subcategories in terms of um, what a protein is carrying out. And we're going to dissect it a little bit. And we'll get into one of those major groups, those being our enzymes. But for this, we're going to talk about protein ligand binding. And I say that ligand, even though I will typically say ligand. Those are interchangeable pronunciations, so don't really get hung up on that. Um, Now, what I want to first address with y'all is that the function of many proteins involves reversible binding of molecules. So just imagine that you have, here's a protein, and here is a molecule that binds to it. That would be a ligand. And so what we can have is where we have a separate protein and a separate ligand. And then we could also have something like this, where we have a PL complex. Now, basically, since Gen Chem 2, one of the fundamental things that you talk about is equilibrium. So this complex forming and this complex dissociating, or associating and dissociating. Now, this big term ligand or ligand, that's our molecule, our smaller molecule that binds to a protein. Ligands can have a variety, or they can be a variety of molecules. They can be proteins themselves, they can be small organic or inorganic compounds as well. Ligand binding is, so this event right here, it is most importantly these three things. It is non-catalytic. Second, it often causes a protein's conformational change. So if you imagine this term conformational, think about that as shape. So the way I will visualize it is, let me go ahead and make an edit on my previous structure, and instead, let's draw my protein a little bit differently. So it's here, and then I'll make it like abruptly or like considerably different. It's gone from this rounded shape to a square shape. Obviously, rounded and squared, not really what a protein's going to do, but that's just my model here. Um, so this often causes a conformational change and it can be regulatory. So what that means is that this molecule, like this protein right here, it can have one function. It can carry out a function. Let's just say that it has functions A and B. And whenever the ligand does not bind, this protein carries out function A. It doesn't matter what that is. I'm just using it as a space filler. But then whenever our ligand binds, then all of a sudden we have this blockage of A. A is not going to take place because the ligand has bound to that protein. Now, the model protein that we're going to use is involved in this process, oxygen transport. Here what we have is a ligand that everyone's very familiar and very thankful for. That ligand is oxygen. Now, oxygen is in a very high concentration in the lungs. So when you do this, our lungs fill with oxygen. There's a high concentration of it in the lungs. Compare that to in my peripheral tissues, in my the the far parts of my body, my toes and my feet. Those are farther from my lungs, and so the oxygen concentration is fairly low. But I do need that oxygen to get there. And so there's a protein known as hemoglobin, which can bind oxygen and take on this form known as oxyhemoglobin. What's a little bit difficult to see here is that we've got a total of one, two, three, four oxygens which have bound to hemoglobin, making this tetraoxygen oxyhemoglobin. Now that is going to go through the arteries, ultimately make it to some sort of tissue, peripheral tissue, and that oxygen bound to hemoglobin is going to be released. That oxygen bound to hemoglobin is then going to bind to this protein known as myoglobin. Myoglobin is going to hold on to this oxygen, and then when the time is right, it will release that oxygen. That oxygen will go to the mitochondria, where we can undergo cellular respiration. And that cellular respiration, well, that's an O2 demanding process. That O2 demanding process will produce this molecule known as carbon dioxide, which carbon dioxide will react with H2O and form this compound right here, HCO3 minus. That combined to hemoglobin, which I'm going to write down as HB. That HCO3 minus will release H2O that will bind to hemoglobin. That HCO3 can then go back into the lungs where it's converted to CO2, and that CO2 can ultimately exit. And you can exhale it. So hemoglobin in this process has two major states, oxyhemoglobin and deoxyhemoglobin, which I'm just going to write HB. And think about HB as hemoglobin as opposed to the 2004 stellar movie Hellboy starring Ron Perlman. Now, myoglobin and hemoglobin are proteins that we're going to use, and we're going to study these two proteins. Myoglobin functions as an oxygen storage and transport protein in the muscles. Hemoglobin functions in an identical capacity in blood vessels. What that means is that they both can bind to O2, they can hold on to it, and they can move it. The difference is that one of them is in a very mobile part of our bodies, and that would be hemoglobin is in the bloodstream. And so it's going to go from one part of the body to the other parts of the body. Myoglobin is pretty stationary in that it's going to remain in the muscles. So myoglobin and hemoglobin are built on a common structural motif. So what that means is that a 3D model of this protein, or a structure of this protein, kind of like an element known as a structural motif, that specific structural motif is known as a globin fold. Now, here's a model of myoglobin shown on the right, which I will write down as on the left, myoglobin right here, and hemoglobin on the right. Now, what I want you to look at when you look at the structure is let's start with myoglobin because it's a little bit easier to process, in part because it's just one polypeptide. Hemoglobin is 4 polypeptides. One peptide. 4 peptides. So when we look at myoglobin, we have A, B, C, E, E, F, G, and H. Okay, so now that's not really a complicated thing to dissect in terms of letters and knowing what they are, but what's a little bit challenging about this is understanding what that means. So all of those letters are to kind of call out specific alpha helices. So secondary structures. So these secondary structures, these secondary alpha helices are found in these different locations. So this would be an A alpha helix, a B alpha helix, C, D alpha helix, E, F, G, and H alpha helix. Now, this is myoglobin. You have all those different alpha helices. And it's really just a matter of, for people who study myoglobin structure, what they're going to do is they're going to say, oh, in the C helix, there is one amino acid and it's protonated, just to use some of the lingo and the jargon that would be used. So then what we'll follow that up with is someone else would say, oh, well, in the D helix, it's deprotonated. Now, when we look at these different subunits, now we're going to switch pages and look at hemoglobin. We see A, B, C, D, E, F, G, and H. If you were to take the structure of myoglobin and basically rotate it and superimpose it over one of those different subunits of hemoglobin, you would see there it's a very strong overlap. So it's almost like when you think about a Venn diagram, you have these two regions where there's not much overlap. This would be something that looks like this. The structures really strongly overlap one another. Now, big structural difference between them is that myoglobin, like I said, is one peptide, and hemoglobin is four peptides. The polypeptide chains, what they do that's very important is they form or they fold around a prosthetic group, which I'm going to highlight right here. That prosthetic group here, 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 and here is a molecule known as heme. Heme holds on to an iron ion. Now, this is another depiction of myoglobin with that heme highlighted. In the center of that heme is an iron ion, and that iron ion is what is responsible for basically coordinating and binding to oxygen.